Thank you for the opportunity to speak to you today. I'm going to give you my perspective about where things stand in development of an HIV vaccine, convince you that we need one, and talk about really two extreme aspects of it. That is, where are we distally in trying to understand the effectiveness of certain regimens, as well as where the next generation of immunogens in the pipeline, and how can we also accelerate this process. So I'd like to just start with perspective of where HIV infection is globally, and how does a vaccine actually fit into this. The UN AIDS report that came out in December of 2017 estimated that there are now 36.7 million individuals living with HIV infection. And of those, 20.9 million are accessing antiretroviral therapy. The fact that much therapy is has been introduced and can be made available to individuals worldwide is a major accomplishment in this epidemic, and does account for lowering rates of new infections as well as deaths each year. However, there is a goal to try to end the epidemic by 2030. And in order to accomplish that, we really need to get to less than 500,000 new infections by the year 2020. And we're really unlikely to make that based upon current projections. So as you can see here, with 1.8 million new cases in 2016, the last data sets that we have really are showing you that we're very unlikely to get there within the next 4 years. Moreover, the amount of resources that are really pouring in to make treatment and prevention efforts available for low and middle income countries is astounding. It's between 15 and 19 billion dollars per year, and that's been the case since 2008, so now for a decade. And so to continue at that rate, just to hold infections down to what we're seeing now, it's really unlikely that we can sustain that type of effort. And so for that reason, we think a vaccine is needed. The second point is that using modalities for prevention and for treatment are being studied carefully now to really understand how aggressively can these be used in order to accelerate the process. And so, as you know, antiretroviral drugs are now being used as pre-infection, pre-exposure prophylaxis, called PrEP, in individuals who are at high risk for acquiring infection, and mostly in endemic areas, but also across the world, as well as in those who are infected. The implementation of antiretroviral treatment in individuals as soon as they are diagnosed with HIV infection. So an interesting study that was published last year by Helen Reese and her colleagues, she looked at, it was a demonstration project, and she was recruiting female sex workers in South Africa at risk for HIV infection. They came in, they were tested for HIV infection, and among those who remained HIV negative, they introduced a study they were going to do to give them pre-exposure prophylaxis, and if they were infected, then they may ask if they would be willing to undergo treatment with antiretrovirals, and to be followed. So the follow-up to date for this report was 12 months. And among those that were enrolled, if you just looked after the first visit, within a short period of time, at this point it was at 1 month. Those who received the pre-exposure prophylaxis, the numbers have gone down to 53% among those who had just enrolled 1 month previously. And by the end of that year, only 22% of that group remained on pre-exposure prophylaxis. And so the attempts to really implement widespread pre-exposure prophylaxis and adherence over a long-term basis is unlikely if the patterns are similar to what is here in an area that is really among female sex workers who are in part really fueling the epidemic. The numbers are better for those who remained on treatment within a year. That was 60%. But still, both of these data sets really highlight the challenges of trying to sort of treat our way through this epidemic. And so for that reason, a vaccine is needed. And so where does that stand now? So really, we only know whether a vaccine will have utility is when we can show that it is efficacious in phase 2B or 3 trials. And among those that have been conducted to date, basically 3 different concepts as underlined at the bottom of the graph. Since 2003, only 3 concepts have been tested. One is a subunit protein with adjuvant of the HIV envelope. One is the Merck Ad5 recombinant vector encoding HIV gene products. And one was a prime boost regimen that was a DNA prime and then adenovirus vector boost encoding HIV genes that represented multiple clades. In this case, none of these 3 concepts showed efficacy in high-risk populations. The only trial that has shown efficacy is called the Thai trial, the RV144 trial. In Thai study participants, there were about 16,000 participants. They were immunized with a canary pox vector encoding HIV genes, gene products, and then they were boosted with the combination of that vector again with the recombinant GP120 protein that was formulated in alum. And those data came out in 2009. As yet, we have no new efficacy data to report, and this is where we are at 31% efficacy. So the importance of that study was, although it still in some ways remains controversial, is that there is the possibility that an HIV vaccine that could prevent HIV infection is possible to make, but we really have a long way to go. And so what attempts are now being made to improve? So we have a number of advances in the vaccine development field that are really helping us to move the effort forward. And let me just very briefly highlight those. A number are in the area of T-cell-based vaccines, trying to induce primarily CD8 T cells as well as CD4 T cells. And the other efforts are around antibody-based vaccines. And the larger effort right now is really focused on the antibody-based vaccines. So for T-cell-based vaccines, probably one of the biggest highlights is the use of CMV vectors, which have been shown in non-human primate models to induce unusual CD8 T cells that have cytotoxic activity that can recognize CD8 epitopes that are presented by MHC class II and MHC, HLA-E. And these have shown about 50% protection among a number of different large experiments. And most of this work has been done by Louis Picker and Scott Hansen at OHSU in Oregon. And these are exciting, and as you know, these vectors are really being now looked at for pathogens other than HIV. But the first clinical trials of these vectors being initiated in Portland now. There's also an approach that really is focused on trying to use gene inserts that are based upon conserved or centralized sequences that are more like consensus sequences, or even mosaics of a large view of all the epitopes that are circulating for CD8 T cells and CD4 T cells. And so these are currently also undergoing early stage studies, as well as there is one now that's just moved into an efficacy trial. The idea of the Mosaic approach was really initiated by Betty Korber at Los Alamos National Labs. The idea is really just to improve the coverage of, in order to recognize more diverse epitopes. For antibody-based vaccines, some of the key things that are helping us to design new immunogens are based upon better knowledge of what the native envelope trimer structure is by cryo-EM and crystal structures. This has really been very innovative and helped us to think better about how to represent the vulnerable epitopes of HIV to neutralization. With this knowledge, a number of new immunogen designs have come forward that represent sequential immunogens that can drive the immune response across a lineage pathway in order to eventually induce broad neutralizing antibodies. And these are both B-cell lineage and germline targeting approaches. And then there's just been the overall identification of even more broad neutralizing antibodies. These have now been made into monoclonal antibodies for clinical use, and they're being tested alone and in combinations. And these offer really promise in use for monoclonal antibody therapeutics, as well as passive immunoprophylaxis. And there is a major study now ongoing for passive immunoprophylaxis to determine if this might prevent infection. So, if we start with the RV144 trial result and think about where are we in terms of extending and improving, the first effort really focused on vaccines that can improve the potency and durability of non-neutralizing HIV envelope antibodies that recognize the V1, V2 region, because this was identified as a correlate of protection in an immune correlates analysis after the outcome of the trial occurred. Based on this, there are now two efficacy studies that are ongoing. One is basically using the same combination regimen as RV144 trial with the ALVAC vector, but it now encodes circulating strains from clade C HIV subtypes that are circulating in the region of Southern Africa where these trials are ongoing. So this is in the HVT 702 study. The other trial is, has just recently started, and those are using the Ad26 Mosaic approach, followed by a GP140 trimer boost with alum, and that was started in November of 2017. So these are now ongoing, and hopefully one of these will improve upon efficacy than what we did see in the Thai trial. Our laboratory and others were involved in pivotal trials to advance these forward, and just looked at the peak responses in these earlier stage trials to determine, what might be important about these regimens and how are they different. In those studies, we were able to show that in terms of antibody responses, they had variable pluses and minuses in terms of inducing this anti-OM V1, V2 antibody responses, really dependent upon the antigen reagent that we were looking at. But the LVAC GP120 regimen appeared to have increased Fc effector, functional activities such as antibody-dependent cellular phagocytosis, whereas the Mosaic at26 regimen appeared to have a stronger anti-OM GP41. So we don't know how important these antibody responses will be, but at least they are giving a different profile. In addition, we looked at the CD4 T-cell responses. We had found previously in the RVN44 trial that 31% efficacy was associated inversely, was associated with a polyfunctional CD4 T-cell response for recognizing HIV envelope. When we look at the two regimens, we see that there is a different quality of the subset responses to HIV envelope. That is, with the canarypox GP120 regimen, we see more IL-4, Th2 type inducing cytokines in contrast to the Mosaic vector approach. And in summary, while the CD4 T-cell envelope-specific responses rates are greater with the Mosaic approach, sorry, I keep hitting the advance button, were greater in the Mosaic approach, we did see that the polyfunctional responses were greater for CD4 with the canarypox regimen. And also the Mosaic regimen was able to induce CD8 T cells, which we did not see with the canarypox regimen. So different patterns of both antibodies and T cell responses, and so potentially we will gain some traction with these approaches in efficacy. Now, I mentioned that other novel approaches are really to now try to either induce broad neutralizing antibodies or to provide immunoprophylaxis with monoclonal antibodies. And so currently we now have one large study in phase 2B that's really across the world in both the Americas as well as in Southern Africa. And then there are already immunogens moving into phase 1 to try to induce these broad neutralizing antibodies. So I want to highlight what we call the AMP study, the antibody-mediated prevention study, that are now 2 phase B studies that are occurring in MSM and transgender in the Americas, 2,700, and then in sub-Saharan African women, 1,500. They're receiving the monoclonal antibody called VRC01, which targets the CD4 binding site. That's a site vulnerable for neutralization, and this particular antibody, one of the first discovered for the CD4 binding sites, in fact, can induce broad neutralization across a large number of virus isolates that are circulating. So this is being infused intravenously every 8 weeks times 10 in these individuals, and we're looking at 2 different doses, and this is ongoing. we wondered and needed to know before initiating the study whether or not this monoclonal antibody could actually reach the sites of HIV exposure. In this case, for these risk groups was in the mucosa, both genital and rectal mucosa. We looked at this by taking biopsies from individuals who had been exposed with this monoclonal antibody and then ex vivo infecting them in an explant model, and then measuring also the concentrations of those antibodies in secretions and in tissue. And what we're able to show is that the concentrations were lower in the secretions and in tissues, particularly the tissues, depending upon whether it was from the female genital tract or from the rectal compartment, and about, in this case, about tenfold lower than what we saw in serum levels. This is just to give us an idea of what that concentration is likely to be once HIV is encountered. In these explants of those who had been infused with VRC01, we could show that, in fact, the tissue with the antibody was able to inhibit viral replication. And in most cases, we never saw replication carrying these experiments out for quite a long time, except there does appear to be some breakthrough in the rectal tissue, and further data also has shown this. We think that it's likely that there are more target cells there, and that it may be that it's that because of that, it's more susceptible to infection. So this study and the active vaccination efficacy studies I talked to you about are really covering many, many sites in sub-Saharan Africa, 8 countries, and shown here. And this has really been a process of trying to large effort to develop a large infrastructure of clinical trial sites that can do these studies. You know, many of these, as I said, IV infusions every 8 weeks, and it's a major effort both for the clinical trial structure as well as supporting the laboratory component with that at each of those sites, as well as we've more recently in 2013 established a laboratory in Cape Town as a sort of reference immunology lab where we can do as many of the analyses as needed for the conduct of these studies. And this is the lab shown here. It's been very well equipped. We were fortunate the Bill and Melinda Gates Foundation provided funding for us to establish this lab, and it's a very busy lab now. Okay, so when will we know anything about these studies? Really not until toward the end of 2020 and going into 2021. So the really timelines are shown here at the end. Providing these are successful studies, we should know something starting in October of 2020 through March of 2021. So we still have a bit of time to wait. So what we have also in place is a framework to institute immune karlitz analysis. So one of the major rationales for doing these studies is that even if we don't hit a home run that would go to licensure, we really want to be able to understand if the vaccine can work, what might be the mechanism. So we have a number of studies, lots of archive samples that will be tested once we really know what the signal is and can develop a case control study. So in the meantime, we're busy with looking at these new alternative viral vectors and prime boost regimens. And just to highlight one is a DNA Mosaic approach that's being led by Bart Haynes, and the studies being conducted in our network, which is headquartered here at Fred Hutch. But you can see there really are still a number of others that we hope can come through that might serve us better than those currently being tested. We're using a Mosaic approach that was described in the non-human primates in 2 articles and developed by Betty Korber. And what we're seeing now is in the first study with this DNA mosaic vector followed by an MVA vaccine is that there does appear to be some enhanced coverage of the envelope epitopes in comparison to those individuals that are receiving just consensus strains or just receiving inserts that just have one natural isolate. And if you look by epitope mapping, this is all still in progress. We are seeing, appears to be that we're seeing more epitopes that are being recognized by HIV envelope than we do with the others. So that hopefully will be helpful in providing coverage for T cells. Then also we're interested in a pipeline of broad neutralizing antibodies. And here the strategies are to try to develop antibodies that can recognize sites of vulnerability on the envelope molecule, and there are a number of these that have been described, even more than what I'm showing here. But really the idea is to be able to eventually mature an antibody response so that we will get high affinity antibodies that can recognize one of these epitopes. So this approach is underway, and immunogens are in phase 1 now. Some are directed toward lineage pathways, and some are really looking at scaffold proteins of epitopes, and a lot of different ways. And then also we're using the native envelope trimers, particularly as ways to boost these initial germline responses. So the key questions that we are trying to address in these first-in-human studies with these BNAB type approaches are obviously, can these new envelope immunogens induce broad neutralizing antibodies? And we think that potentially an early step in that process that we hope we can at least see is whether or not they can neutralize the autologous strain that's represented in the immunogen, because most of these are really hard to neutralize strains. So autologous tier 2 neutralization. And do we activate the relevant naive B cell repertoire? So a lot of sequencing of naive B cells and then looking at the pathway of response. Can these responses be boosted and shaped by enhancing somatic hypermutation and affinity maturation? And then what role do CD4 T cells, particularly T follicular helper cells, have in shaping the B cell repertoire to induce broad neutralizing antibodies? So major efforts in the field are going toward addressing these questions. In order to do that, we're no longer for carrying out phase 1 studies that are sort of recruit shoot. We're really trying to interrogate the immune system in a very systematic way with approaches that really can help us understand what is happening in the germinal centers of draining lymph nodes, and then how is the affinity maturation occurring, and then what are the long-term durable effects. So it's a whole host of very intensive analyses in these phase 1 studies with a number of long list of activities that we're doing to really look at these approaches. So one effort that is underway now that seems to be working well is to try to interrogate the draining lymph nodes from the site of the injection of the vaccine. And we've piloted this using a licensed vaccine, in this case hepatitis HPV vaccine. And in doing that, we give the vaccine, and then after about 3 weeks, we aspirate lymph nodes from the draining site, either the axillary lymph nodes if it was an immunization in the deltoid muscle, or the inguinal lymph node if it's from vastus lateralis, quadriceps muscles. And then we look at both the CD4 TFH cells as well as the activated B cells that are recognizing HPV. We've now got nearly full in this study, and we can very easily pick up the germinal center TFH cells as highlighted here and some of the phenotypes of those cells, and can do sort of deep probing with single cell RNA-seq to try to understand the heterogeneity of these responses, as well as look at the B cells and how they are maturing over time. So this is a nice approach which we're now will be using in the HIV immunogen study starting in 1 month. So I think we're ready. And then the final thing that we're doing in this area is try to see how can we pair novel adjuvants. As you saw, most of this was with either alum or with MF59. Are there newer ones that could prove more potent or give us a different quality of response? So we're really now going to look at these native envelope trimer immunogens, as shown here by EM, and formulate them very carefully with novel adjuvants targeting different TLR pathways, and see if we can improve those responses than what we've seen with alum. And so those studies will be starting early next year. So just looking forward, important areas that the field now can and will address while we're waiting for those efficacy results to come out are, can we identify new vectors that induce unique or preferred responses over the Ad26 and ALVAC vectors in these prime boost regimens? We can assess whether novel adjuvants are safe and improve the quality and durability of antibody responses. We can establish whether or not broad neutralizing antibodies given intravenously or ultimately sub-Q in combinations at some level can protect against HIV infection. And we can determine if novel envelope glycoprotein immunogens can induce broad neutralizing antibodies. And it's really probably the latter, which we feel is the holy grail to eventually get there. Potentially the shorter term solution for immune-based prevention is with the monoclonal antibodies. But we do think that active vaccination over time is really what we need for long time durable immunity to end the HIV epidemic. I'll stop here and just very briefly highlight key individuals involved in this, a host of people in my lab working on this project shown at the top here. We have this large international network that you saw pictured that's led by Larry Corey and Glenda Gray and Scott Hammer, an amazing statistical group at the Hutch who work with us, led by Peter Gilbert. And then our funding primarily comes from the National Institutes of Health, the Division of AIDS, as well as from the Bill and Melinda Gates Foundation, and some from the South Africa Medical Research Council. Thank you.
So we'll do things a little differently now. So we'll take a couple of questions, and as we're doing the Q&A, we'll set up a panel discussion so we can go directly to lunch without a break right now. So just to get back on track. So are there any questions for Julie?
Hungry. Well, I think the CMV vectors, if they can really be clinically applied, have promise. But the key thing that has not been addressed with those is why there's, just always only protection in about half of the monkeys. So it really hasn't been nailed why that is the case. And until that is, and we can then do something about that, I think that's going to be an issue. With the, I think the broad neutralizing antibody concept with these immunogens I talked about at the end, it's really been shown successfully now in knock-in mouse models, humanized, that, that there is a path and it can work. But, it's really rigged for the system, and whether we can get there in people and that they can really be recognized and driven correctly down the pathway, I think is just going to be a hard thing to do. I think that, for the monoclonal antibodies, I think they're the short-term solution, and that we will probably get a home run on those, particularly if we can give them in concentrations that can be given subcutaneously and in combinations, and we don't get resistance. And that could be done for the period of time when individuals are at risk for exposure. But I think that either of these vaccine-type approaches for both T cells and monoclonal and antibodies are going to still take decades.
So, one more question. What about the T cell vaccines as opposed to the B cell?
Yeah.
Is there a clear statement there?
Yeah, there's always these camps. There's beginning to be more talk about, having these camps kind of come together. It's probably still going to be a combination of those. I just don't see that one could really do the job completely. Yeah, thank you.
Can I ask the speakers to come on stage, and we can
Thank you.
thank Julie in the meantime?